All right, we can get started. For those of you who don't know me, I'm Craig Jordan. I've had the pleasure of taking over as one of the moderators for the DOM series this year as Sean Colgan has stepped down. So it's a pleasure to introduce our first speaker this morning, Dr. Christopher Evans from the Pulmonary Division, also with a joint appointment in the Immunology Department here at the University of Colorado. Chris did his MD PhD at Johns Hopkins University, followed by postdoctoral work at the Texas Medical Center involved with both Baylor and MD Anderson. From 2005 to 2011, he served as an assistant professor at MD Anderson, followed by his recruitment to the University of Colorado in 2011, where he has risen up to the ranks of the associate professor level. Chris has a really wonderful academic career thus far. He's been very active in sort of all aspects of what make a great academic noticeable, very well-funded speaking engagements really all over the country, multiple mentees and educational opportunities, as well as a robust academic and research program. So obviously today he has the provocative title telling us a little bit about unexpected roles of mucus. Chris, thank you for being here.
Thanks so much. Yeah, I hate putting in these jokes about flavors when people are eating lunch and thinking about the taste of mucus, but it's hard to restrain myself. This is a picture from the top of Mount Bierstadt. Went on a hike up there a couple years ago, and it was a really pretty picture. Out here in the distance, the thing that looks like a whale hump, that's Pikes Peak down there. So it was a pretty day where it was cloudy over Denver and sunny on that side of the ridge. So since this is a mixed audience, I thought I'd start with the Captain Obvious picture here, which is to tell you what the lung is for primarily. It's gas exchange. So the intake of oxygen, the release of carbon dioxide, and air passes through these tubes of the tracheobronchial and bronchiolar airway generations and down to the alveoli. And what I'd like to highlight here is at the alveolar level, this is an extraordinarily thin layer here. So it's less than 1 micron thick. You can see a blood vessel with a capillary in it here. So the diffusion of oxygen across here is maintained by how thin and delicate that surface is. But it becomes more susceptible to injury than one might like. So the airways have developed numerous ways of maintaining defense against foreign objects, as well as controlling inflammatory responses that may disrupt homeostasis. So to that end, whether we're outside in nature or in the city, we're exposed to tons of microbial insults and particle insults. These include allergens, microbial agents, as well as pollutants. And just to put these in perspective, if you think of a tidal breath, just for round numbers, half a liter per breath, and this number of breaths per minute, and this number of minutes per day, you take about 12,000 liters of air into the lungs on a daily basis. So that's a pretty massive volume. And when you take into account that within that 12,000 liters, there's approximately 10 to 20 million particles per liter. And if you can do the math and extrapolate per day, there's 100 to 200 billion particle exposure to the respiratory tract on a daily basis. A fraction of these are microbes, and again, you can kind of see the math as it works out here to hundreds of thousands to millions of microbes that can end up in the airways in any given period of time. So to that end, the lung has developed a system of primary and secondary innate defense mechanisms that can be used to maintain homeostasis. And while they do that, though, to do so without causing massive inflammatory responses and exaggerated responses that might somehow damage the tissue and compromise the host. A first layer of defense is the epithelium. And as that primary barrier, the epithelial barrier is shown here in a mouse trachea, this freeze fracture SEM. And you can see that the surface of the epithelium contains this blanket of mucus. And these cells right here are ciliated cells that project up into the mucus and allow for clearance of things that become trapped in that mucus blanket, where they can then be eliminated by either expectoration or swallowing. Again, another tasty lunch subject. As particles impart into the lungs, the other stereotypical mechanism of defense that's been, that's again, in all the textbooks, is the alveolar macrophage. So these are just pictures of alveoli down here, and you can see a macrophage there in that picture of an alveolar duct. And the alveolar macrophage also helps to provide defense in this sense against very, very small particles that don't impact upon airway surfaces and get down deeper into the lungs, as well as particles that might just collect there over longer periods of time and bacteria that may grow. In this case, the alveolar macrophage is very capable of eating, phagocytosing, and killing organisms And in that sense, it's also very restrained from inducing inflammatory responses upon exposures. The classic example I showed you there, the alveolar macrophage, is again, what's in all the textbooks. What's a little bit less appreciated is that the macrophages are present all over the airways. This is a rat trachea. And you can see airway epithelia here. You can see ciliated cells mixed in with non-ciliated cells. And these white specks here, these are macrophages. And you can see on this portion over here, the macrophages are covered in a mucus blanket. So there's a physical interaction between the two systems, the primary layer of mucus and a secondary backup line of defense, the macrophage. And we're interested in exploring how these two interact, and in that sense, how they can maintain homeostasis, but how they can protect against pathophysiological situations. And again, when these things go wrong, a lot of damage can occur. So when mucus hypersecretion occurs, such as in asthma, airway plugging and obstruction is a phenomenon that's well known. I used asthma as an example, but you can also see it in COPD, cystic fibrosis, and in other diseases. And then as a second layer of this, though, when mucus is not cleared properly or is produced in a dysregulated fashion, inflammatory responses that usually require the mucus layer to help them be under control and to be cleared out can be exaggerated. Bacteria may accumulate, as seen in CF or COPD, and inflammatory cells may be altered in their ability to be eliminated from the lungs. And in that said, in the chronic settings, you can have a situation in which mucus dysfunction can also cause long-term structural remodeling in the lungs. And so in this situation, what's an example here is in pulmonary fibrosis, with a diagram here showing that with the excessive mucus that's being produced in very small airways, there's a prevalence for fibroproliferative changes that can occur. So in this sense, what I've highlighted here is that there's a balance that needs to happen for homeostasis and pathophysiology, in which mucus and the particles are either cleared or the airways get plugged. The lung is either defended or becomes chronically inflamed, or situations occur in which it can be repaired and homeostasis restored, or you can have remodeling, and remodeling in this case can result in devastating consequences. So how do we address this? You know, we've talked about mucus and made the off-color jokes about it, but really what it comes down to is that there's a handful of genes that encode the mucin glycoproteins that are present within airway mucus. The ones that have been shown to be expressed in the lungs, at least at some level, are MUC2, MUC5AC, MUC5B, and MUC19. This is mouse lung, it's qPCR from a mouse lung. And you can see that MUC5AC and MUC5B are the predominant species present. This is a log 2 scale, so these are many-fold higher than each of these less abundantly expressed isoforms. But you can see that at baseline, MUC5B is predominant, and MUC5AC is lower, but under inflammatory conditions, in this case in an asthma model, MUC5AC can be induced. And it turns out that this 40 to 50-fold induction of MUC5AC puts it to equal levels with MUC5B. So you have two mucins, 5AC and 5B. And based on that, we've decided to explore them using genetic tools to be able to address how they function. So in the baseline state, as I showed you, MUC5B is produced, MUC5AC is low. And in the allergic setting, they're both present. There's a little bit of an increase in MUC5B. It's about 2-fold, which is consistent with what you see in the qPCR before, but a robust induction of MUC5AC. Based on this, we took MUC5AC knockout mice and exposed them to an allergen. And we tested whether or not they had an airflow obstructive phenotype that's consistent with asthma. So in a wild-type mouse, the plus line shown here with the black dots, the allergic mouse shows exaggerated responses to methacholine. Methacholine is an agonist that causes bronchoconstriction but also causes mucin secretion. And the methacholine responses that you can see in the allergic mouse are potentiated over the non-allergic wild-type mouse. But you can see that the MUC5AC knockout here, this minus in pink, is protected from that exaggerated response to methacholine. So it's protected from airway hyperactivity, consistent with the protection from the asthma phenotype. And it turns out that this protection came with a restoration of airway opening and a loss of airway plugging that's seen in the wild-type mouse. In this same context, though, inflammation was not affected. Right? So this is, if we look over here at this BALB-C AOE column over here, the numbers of eosinophils, which are 0 in a saline challenge mouse, are indistinguishable in the knockout mice. We did the same experiment in multiple strains of mice and with multiple allergens. But I just wanted to point out that MUC5AC does not appear in this setting with a chronic allergen exposure to affect inflammation and the control of inflammation. Instead, it seems to play a bigger role in obstruction, so more of a physical role than an inflammatory role. So this starts to get us thinking about how these things function. So we've talked about which genes are there, which ones might be involved in a pathophysiological response. But when we really want to get down to the nuts and bolts of how they work, we have to think about them at a structural level. So these models up here are MUC5AC and MUC5B apoproteins. They have von Willebrand factor-like domains in their N and C termini. These form disulfide bonds that cause these things to link together in very large polymers. A single dimer of this is about 1 micron long, and these things can make 10 to 100 mer length pieces. So they can be extremely large molecules in the airway surfaces. They're held together by disulfide bonds. There's another area of internal disulfide bonds that we don't really know exactly what they do. It's thought that they help regulate the mesh pore size of mucus gels. But then the other business end of the mucin, and really what makes it different than almost any other protein in the body, is its heavy glycosylation. So it's serine and threonine residues. It gets core glycosylated. These glycans can be elaborated with the addition of glycnac and galactose residues, and termini that include sialic acid and fucose residues as well. So you have some specialized sugars that can get added onto it. I'll come back to these later on in the talk, but what I'd like to do right now is to focus on that first end here, the disulfide end. And in our asthma model, we asked the question, well, if you form these big plugs and you have this really big molecule, what happens if you can break it up? And so we collaborated with a group that was a spin-off company started by Ric Boucher at University of North Carolina. And we tried using a mucolytic agent that this company had generated. So Leslie Morgan, who's a graduate student working in my lab, has taken mice that are exposed to an allergen, in this case AOE, and that stands for Aspergillus oryzae extract. And they were either exposed to the allergen and then methacholine, and then treated, in this case, with saline. So this mouse was challenged and inflamed, but it didn't receive this drug. And the drug has a sort of jargony name, so it doesn't really help to understand that at this point. But the drug was given in pulses at each of these points between methacholine doses that were being administered IV. And you can see that the mouse that had been challenged but not received the drug had an exaggerated response to mice that were not challenged. And the mice that received the mucolytic agent were protected from airway hyperresponsiveness to methacholine. In fact, their levels resembled that was seen in MUC5AC knockout mice. So she repeated those studies in this case with the IV methacholine challenge. And it turns out that the protection that she was seeing with this mucolytic agent was associated with a disruption of mucus polymer size. So as I was saying before, the very high molecular weight mucin polymers, they can be seen here. This is an agarose gel that's run overnight. Most of the material that you're looking at here is migrated less than a centimeter into the gel. And so these very large proteins can be seen in mice that were treated with vehicle, in this case saline. But mice that were treated with the drug show disruption of the mucus polymers. And that's consistent, again, with protection from airway hyperresponsiveness. And Leslie's in the process now of going through and doing quantitative histology to look for plugs in the airways and to complete that portion of the study. So we talked about MUC5AC and its role in plugging. But what about MUC5B? You know, it's sitting here at baseline at very, very high levels. And what we wondered is how does this function and what does it do? And MUC5B is a protein that's involved in mucociliary clearance. So when we look at MUC5B knockout mice and compare them to MUC5AC knockout mice, there's a significant impairment in mucociliary clearance function. It's defined here as MCC. So in this case, particles are instilled into the lungs, and we looked at their elimination over time. And there's a dramatic impairment in the MUC5B knockout mouse. That loss of clearance is associated with an accumulation of bacteria in the lungs. So in this figure here, what I'm showing is mice that were sacked at 3 months or 6 months of age, and we called them healthy in that they were not dead. And in this case, they showed that they had increased accumulation of bacteria in the lungs. But then we did check on mice that were becoming mostly dead. We were able to see that there was even a higher level of bacteria that were found in the lungs. And so in this case, we have an expansion of bacterial colonization or infection in the airways, and that was consistent with a loss of life phenotype. So in this case, the knockout mice are shown here in red, and you can see over time they petered off. But when we put mice on antibiotics starting at weaning, that's the green line across here, we protected them against the lethal phenotype. Turns out that these associations that we're seeing here, or in this case the causative nature of the infection, is associated with chronic inflammatory settings. And again, it makes a lot of sense considering that you have a bacteria accumulating in lungs. What we see is that over time that there was increased macrophages, neutrophils, and other cell types, including even a few eosinophils, present in the lungs of the mice, even those that survived out to longer lives. So we started to think about how could there be a functional link between these. So there's an association between the numbers, but what happens to the cells themselves? And one of the most striking things that we'd seen was just a simple cytospin, where we were able to look and see that by 3 months, and in this case in the surviving mice, by 12 months, you can see that the macrophages don't look good. They look, in this case, angry, or in this case, exhausted. And that phenotypic morphologic finding was associated with loss of function. So their phagocytic activity was decreased, and it turns out that they were also apoptotic in many cases. So there was a loss of macrophage function and survival in this case as well. So we're at the point now where we have mucociliary clearance and we have a complex phenotype. We have a mucus blanket. We have mucociliary clearance from a distal to proximal airway unit. We have a molecule. We have sugars. We have a lot of complex phenotypes, and they're really hard to explain how you can kill a mouse in this case. And it doesn't really help to try to think of it at a complex level. And so what we've done since then, and this is partly inspired by a talk that Ruslan Medzitov gave here a few weeks ago, or a few months ago now, in which he said, well, take this whole complex system and break it down like an engineer would. And what he said was think about it as a machine. So you have a machine with a set point, as well as controller, actuator, and some sort of control object, so something you can manipulate, and that's what helps you regulate output. And if this control object feeds back to the controller through a sensor, you have a way of controlling the system and regulating output. Again, at any point along the way, though, if you disrupt these components, you can break things out. So taking this analogous setting, what I've done is said, okay, well, let's look at it in this case. So we have homeostasis, and we have defense. Defense could be disease if we drive any of these wrong. And in this case, we have our barriers that are our ways of regulating the set point. We expose mice to challenges, or they're challenged by their normal environmental stimuli. And I said that the control objects in this case are the two mucins that we're looking at, MUC5B and MUC5AC. And they're involved in the potential to be able to control and regulate the resolution of challenges to maintain defense. So you can go in this direction and have defense, or you can go in this direction and maintain barrier or phagocytic function. So in that setting, then, you can have a situation in which you have the absence of inflammation. So you have your barrier, mucociliary, and phagocytic defenses. You can have a transient response, which is really what you want. You want this thing to come in and go away as best as you can. You could have a situation in which you chronically challenge these and you start to overwhelm the system, but the system figures out how to tweak itself and tolerate a chronic exposure. And then you can have a situation in which any of these components fail, resulting in disease. So that said, looking at the mucins, in this case what we've done is to take and develop a improved model of MUC5B deficiency, this using a conditional knockout. And we used a lung-specific knockout, and we're able to isolate macrophages from them and perform microarrays on them. And lo and behold, there's a whole host of microbial or antimicrobial, but as well as inflammatory markers that are differentially expressed in the macrophage. This really doesn't tell us a ton specifically, but it does tell us that there is a quantitative and appreciable difference between the wild type and the MUC5B null setting. So in the absence of the mucin, you do reprogram macrophages in this manner. So coming back then to this model, I moved away a bit from the disulfide components because it doesn't seem to make a ton of sense if we're going to look at a direct interaction. It could make a sense in terms of mucociliary clearance, the removal or elimination of dead cells and things like that. But what we started to hone in on is the sugars. And in this case, we're interested in a particular family of sugars, and that's the sialic acids that get attached to galactose in the termini. And the sialic acids come in two major linkages in non-neuronal tissue, so the alpha-2,3 and alpha-2,6 linkages that are depicted here. And they can be detected using lectins as well as other higher resolution assays. But in this case, using the Machiamorensis lectin MAL2, what we're able to show is that MUC5B is selectively sialylated in alpha-2,3 linkage. So we have an absence in the knockout mouse here of sialo detection. but it's still present in MUC5AC knockout mice. MUC5B is there, MUC5B is not there in the knockout, but you can see that in the presence of MUC5B, you still have sialylation in the MUC5AC deficient setting, showing that MUC5B is selectively and somewhat specifically sialylated in this setting. And the consequences of that is that alpha-2, 3 sialysides are actually important regulators of inflammation. And it turns out that there's a family of proteins called SIGLEX, which essentially stands for sialic acid binding protein, so it's sialic acid immunoglobulin-like lectin receptors. And these SIGLEX are able to bind preferentially to different conformations of sialicide residues. And in this case, SIGLEC-F is selected for MUC5B. And it turns out that the ability of MUC5B to bind to SIGLEC-F drives eosinophil apoptosis. So if mice were treated with IL-13 and eosinophils were isolated, we were able to show that in the absence of MUC5B, there was a loss of eosinophil apoptosis in the lungs. In an in vitro setting, if you just purify eosinophils from the mice and you add mucins purified from tracheal epithelial cell cultures, you can also induce apoptosis. So through a gain of function and loss of function, we're able to show this. And in the in vivo context, we've gone further and explored other asthma phenotypes in them. And, I'm giving a talk tomorrow for the pulmonary RIP, and I'll spend more time on that. But for the sake of today, I want to come back to the macrophage. And the macrophage is the other major expressor of SIGLEC-F in the mouse. And it turns out that this expression is linked to inhibitory signal transduction pathways, including phosphatase pathways such as SHIP. And the ability to do this, we hypothesize, can control inflammatory responses, as well as possibly control phagocytosis. We're going to concentrate on inflammatory responses here. I'm talking TLR, in this case, is a generic term, but we will be using LPS to test macrophage responsiveness in experiments I'm going to show. And the idea here is that the control of homeostasis involves a MUC5B SIGLEC-F signaling axis that becomes dysfunctional during inflammation, but is then required for restoration and resolution of lung health. To that end, if we get rid of the ligand, in this case using our conditional knockout mouse and using a temporally regulated version of it, we can show that when we induce a CRE, in this case a tamoxifen-regulated CRE, we can see that over time we can deplete the lungs of MUC5B, and in that same setting, neutrophilia starts to occur in the lungs spontaneously. Again, so they start losing MUC5B, they are losing mucociliary clearance probably, we haven't measured it. They are possibly being exposed to more microbial antigens that live around longer or endogenous particles. But in this case, at least in a phenotypic sense, there's a consistency with the loss of the ligand and the gain of inflammation. And in that same setting, if we get rid of SIGLEC-F, we also see that there's a mild but spontaneous neutrophilia that occurs. So again, this is in the homeostatic setting. And so this is work that we've been doing collaboratively with Bill Jansen, and he and I just got an R01 funded to take this further. So a lot of the data I'm going to show today is preliminary because it had been used up until now as grant preliminary data. So in order to explore these more mechanistically, what we do is we take macrophages from the lungs of mice, throw them in a dish, and then stimulate them with LPS. And think of it sort of like an AHR type of challenge in an asthma clinic where somebody inhales methacholine and you see what the responsiveness is. If the macrophage is trigger happy, if you put LPS onto it, it's going to respond more than a macrophage that's at rest. So in this setting, what we can do is we can take macrophages, put them in a dish, leave them sitting for half an hour to allow them to selectively adhere, and then stimulate them with LPS for 4 to 24 hours. And we can also mix in inhibitors and other components as well. So in the setting of MUC5B absence, what we find is that if we take the MUC5B conditional knockout mice, throw them in a dish and throw LPS on them, they're trigger happy. So they release more IL-1, KC, and IL-6 than wild type. And also there's an overexpressing line that David Schwartz's group made that we also threw in there for good measure. And it turns out that they're suppressed somewhat compared to the wild type. So there may be a level of protection conferred by having a lot of MUC5B around. In this same setting, if we take wild-type macrophages and then we throw a cross-linking antibody that can cross-link and activate SIGLEC-F, we can suppress LPS-induced IL-6 and IL-1 beta production. So we have a loss of function and a gain of function showing at both the ligand and the receptor level that there's a pathway that can be utilized between MUC5B and SIGLEC-F to control homeostatic programming of the macrophage. So what happens during inflammation? If we put LPS into the mouse, it turns out that the knockout mouse has macrophages that show exaggerated LPS responses. In this case, I'm showing IL-6 and TNF. And if we look at mice that were treated, in this case, with an activating cyclic F antibody in vivo, we found that there was at least a mild suppression of neutrophilia. It's significant, but there's so much other things going on in terms of the epithelial-derived components to inflammatory responses that it doesn't surprise me that you wouldn't turn everything off. But again, if you live and die by the p-value, it's something to put in a grant. As we look at this further, we're growing knockout mice that breed poorly and trying to really tease these out and do this in a more robust fashion. But the other part of the situation that we're interested in is the resolution of inflammation. So as inflammation is induced, there's a peak period of neutrophilia that occurs within 1 to 3 days, and then macrophages that are resident cells are shown in the lungs here. They kind of bump up and then restore back to baseline. But there's a whole recruited monocyte population that comes into the lungs. And Bill's group has shown that you can characterize these by CD11B and CD11C expression. In this setting, the CD11C positive recruited cells are more pro-inflammatory, and it turns out that they lack cyclic F. So when they come into the lungs, they have very little cyclic F. As they arise into the lungs and sit there over time, they do start to express it a little bit. And it's thought that perhaps, and again, thinking in terms of a hypothesis, it's not encumbered by the presence of actual data, but it's thought that this may be a mechanism by which monocyte recruitment is turned off. And that may be a way of inducing apoptosis in them. Again, those are all experiments that we proposed and are getting ready to do now. And so the last phase of this then is what are the roles of these guys and the resolution of inflammation? And so if we look 6 days after exposure to LPS or microbial challenge, we find in the absence of MUC5AC, we have high levels of inflammation. And if we put in this cyclic F activating antibody, we can suppress inflammation and help to bring homeostasis back a little faster. So I'm going to end this portion here, this last little bit here, by just saying that there is a human correlate to everything that we're doing here. So this is a human airway, and it's labeled here. Adrian Stefanski did this. It's labeled here with that MAL2 lectin, and you can see that MUC5B and alpha-2,3 are closely linked. If we use another lectin to look for alpha-2,6s, you can see that they're separated. So alpha-2,3 sialylation of MUC5B is a characteristic of human bronchiolar airways. And Siglex, in this case macrophages, this is work that Bill and Leah did. They express Siglec 9, which is our top candidate Siglec for the human alveolar macrophage. So coming back to the system, what I propose here is that MUC5B plays an important role in resolution through its interaction as a barrier and regulator of phagocytosis. And so I'll end up with the model there. And then I'm going to end with a very final shout out here to make a room for Sean here, which is that this property that we're looking at in the lungs is shared in the gut. So I talked about the mucin sugars earlier. It turns out fucose and sialic acid are both the sugars that terminate specialized mucin glycoproteins. Microbes use the sugars that are on there. So they use fucose in this case. And this is work from Jeff Gordon's group, showing that fucose sensing by Bacteroides theta iodomicron is used to control the expression of a whole host of genes. I think one was named by Marvin Schwartz, that regulate quorum sensing and the growth of competing bacteria in the gut. So that's one way in which this is used. And the other is that a similar pattern to what I was just showing you with the macrophage in the lung is utilized by MUC2, the intestinal mucin, through fucose and other residues that activate and control dendritic cell tolerance-inducing properties. So that it appears that this is a consistent and conserved pathway for regulation between mucins and phagocytic barriers. So on that, I'd like to end and thank the people who did all the work. This was us at our lab retreat last year. There was a lot of business discussed at the time. And I'll leave it to questions and make room for Sean here. It is not, and I think it's a good question. So a lot of pathogens have a lot of these sugars on their surfaces already, so whether they compete or somehow interact, it's not clear. They also have a lot of lectins for the mucin, so it may be bug dependent. I try to avoid the microbial ecology as much as I can, just because it's going to change bug by bug. But it is a good question. Okay, so we have done a recent study, and I didn't bring any of the slides here, but we've taken MUC5AC mice and tested them in both KRAS and urethane models of lung cancer, and find that in both settings the absence of MUC5AC is protective. And it appears that there's something going on with the macrophages, at least in numbers. We haven't gone and phenotyped the macrophages, but it's something we'd like to be able to do. But also the change in mucin expression in that setting could affect other intracellular components, such as ER stress and things like that, that might be affecting the way the adenocarcinoma cells grow. So that said, the background is that adenocarcinomas are pretty good producers of MUC5AC. There's some human correlates to go with it, and that MUC5AC expression levels correlate with survival negatively in the case of patients who have a KRAS mutation and lung cancer. They show worsened 5-year survival and disease-free survival compared to those that have lower levels of MUC5AC production. But all bets are off in EGFR mutants. It's MUC5AC independent. Yes, and I'd say the gut, the papers that I showed one figure of at the end there, the control of that is interesting, and that takes place in the small intestine, where in the presence of MUC2, when you expose dendritic cells to foreign antigens, ovalbumin as well as LPS and other pro-inflammatory factors, in the presence of MUC2 and specifically through its glycans, that causes DCs to tell naive lymphocytes to become Tregs. And so that's thought to be one of the main mechanisms that keeps you from becoming allergic to most of the food that you take in. And so some sort of dysfunction in there could play a role in food allergy. That's certainly where the people who are doing that are going. Yeah, sorry.
Unanswered questions later.
Actually, I have to go to pulmonary fellows.
Well, you know how to find them.
Yeah.
So it's why don't you give it a shot? Yeah. So it's a special pleasure to introduce Sean Colgan. Sean is, I think, probably known to just about everybody in this room. Has really been kind of a senior statesman for research in general on this campus. He's been at the University of Colorado for the past decade. He came here in 2006 and founded the Mucosal Inflammation Program, which has grown into an incredibly robust and respected activity. For the past 6 years, he's served as the vice chair of basic research for the Department of Medicine, so has been involved in touched all your lives in ways you may or may not have known about, but he's been on every committee and every advocacy group to support research on this campus, and so it's particularly nice to be able to welcome him today. He's also, of course, had a very robust research program, very well funded for his entire career. I won't go on with the accolades, but he's had a really marvelous career, first at Harvard for over a decade before coming back to Colorado, which is actually his home. John, nice to have you here.